Distinguished Nobel laureates, honored guests. It's a great pleasure and privilege to welcome you all to the 2024 Nobel Lecture in Physiology or Medicine. My name is Annika Östman Wernersson, and I'm the president of Karolinska Institutet. During the Nobel Week, we pay tribute to contributions made by exceptional individuals whose discoveries, in the words of Alfred Nobel, have conferred the greatest benefit to humankind. These are words that remind us of the profound impact that science has on our lives. Today, I'm honored to extend my warmest congratulations on behalf of Karolinska Institutet to our new Nobel laureates, Professors Victor Ambros and Gary Ruvkun. Their groundbreaking discovery of microRNA and its role in regulating gene activity is a hugely important breakthrough. This year's Nobel Prize in Physiology or Medicine highlights the importance of curiosity-driven research. The discovery of fundamental new mechanisms of gene regulation would probably not have been achieved if goals for the research have been predetermined. I very much look forward to learning more about your findings and how they affect our deeper understanding of the biology of our bodies. Now, I will give the floor to Professor Anna Widell, member of the Nobel Assembly, who will further introduce this year's laureates. Thank you.
Esteemed Nobel laureates, dear colleagues and friends, dear students. On behalf of the Nobel Assembly, I welcome you all to the Nobel Lectures 2024. I have the honor and great pleasure to introduce Dr. Victor Ambros and Dr. Gary Ruvkun, who have been awarded this year's Nobel Prize in Physiology or Medicine for the discovery of microRNA and its role in post-transcriptional gene regulation. The flow of information is central to life. How different organisms perform the critical functions they need to develop, thrive, reproduce, all depend on a basic set of instructions that resides deep inside our cells in the form of genes that are made of DNA containing the code of life. How then is the flow of information controlled such that each specific cell type ends up with exactly the right combination of proteins at each specific time, despite the fact that the basic instructions are the same for all? Many parts of this flow of information have been understood thanks to previous groundbreaking discoveries. The central dogma of molecular biology states that DNA is transcribed into messenger RNAs that carry information for translation into proteins, which perform the specific functions needed for the organism. The central dogma of molecular biology is classic textbook knowledge, taught in schools around the world to students from a relatively young age. We all know it. This year's laureates, Victor Ambros and Gary Ruvkun, have conceptually changed the picture by adding a completely new level of understanding to this classic problem. They both studied heterochronic mutants of the tiny worm C. elegans. These are strains of worm that display changes in timing of development. The focus was thus on peculiar-looking worms, anticipating that they could provide fundamental biological insights. Two particular mutants are at the center of this story. An egg-laying defective worm had been identified. Victor Ambros identified another mutant as having opposite timing defects. Ambros and Ruvkun together managed to isolate the gene, a painstaking task with the technology available at that time. Ruvkun then demonstrated that deletions in the gene's untranslated region affected protein abundance in some mysterious way. Ambros, on the other hand, embarked on the huge task of cloning the first gene and made the surprising discovery that it encoded a tiny RNA without protein coding capacity. Dr. Ambros and Ruvkun compared their results, and as we will hear, this resulted in a dramatic discovery moment when they realized that the tiny RNA was capable of astonishing things never heard of before. Dr. Ruvkun later made an additional dramatic discovery leading up to the realization that tiny RNAs are widely spread all through the animal kingdom, defining the class of microRNAs with thousands present in the human genome. Victor Ambros and Gary Ruvkun thus unraveled a completely unexpected mechanism for how flow of information is regulated in our cells. MicroRNAs determine which of all messenger RNAs are translated into proteins, thus exerting a fine-tuning control station that operates differently from all previously known modes of gene regulation. By working in networks, they allow for combinatorial effects, enabling systems-level actions. In complex life, myriads of complicated biological processes are active simultaneously. All of these have to be precisely regulated without interfering with each other, while also compensating for changing environmental conditions. How is all this possible? Shouldn't it all be chaos? The discovery of microRNAs with their systems-level mode of action does make it a bit more conceivable. We are going to hear an exciting story by two remarkable individuals. Both showed a strong interest in science already from a very early age. As boys, they were fascinated by astronomy, growing up during the early days of space travels. Today, they are passionate scientists who have devoted large parts of their lives to curiosity-driven research, characterized by creativity, open-mindedness, technical ingenuity, strong perseverance, and data sharing, leading up to a fundamental discovery with great benefit to humankind. We now know that microRNAs play important roles in most physiological processes and are involved in a range of human diseases. On behalf of the Nobel Assembly, it is now my great pleasure to introduce the Nobel laureate Victor Ambros. Victor Ambros was born in Hanover, New Hampshire, USA. In 1975, he received a Bachelor of Science with a major in biology from the Massachusetts Institute of Technology. In 1979, he received his PhD from the same institute after studying poliovirus genome structure and replication with David Baltimore. Dr. Ambros then joined the laboratory of Robert Horvitz at MIT as his first postdoctoral fellow, remaining there until 1984. He then took a faculty position at Harvard University. Between 1992 and 2007, Dr. Ambros was a professor of genetics at Dartmouth Medical School. In 2008, he joined the faculty at the University of Massachusetts Medical School. Dr. Ambros is now Silverman Professor of Natural Sciences at the University of Massachusetts Chan Medical School, Worcester, Massachusetts, USA. Dr. Ambros, we are very much looking forward to your Nobel lecture entitled A Marvelous Unfolding Story of MicroRNAs. Please join me in welcoming Dr. Ambros onto the stage.
Thank you so much. Thank you. Thank you. Thanks to everybody for being here. I hear it was standing in line was a pain in the neck, but glad you were able to come. I want to thank everybody who's out there who's ever contributed in any way at all to me being here in this moment. And I'll try to mention some of those people as I go along. I bring you greetings from Worcester, Massachusetts. I'm going to tell you a story that basically is about a story of a paper that we published in 1993. So this story is about more than 30 years old. And the individuals who did the work are in this room. That's Rosalind Lee and Rhonda Feinbaum. So this talk is dedicated to them. First, how did I get here? All right, as you heard, I was born in Vermont. I think I started out as a scientist. I don't remember not feeling like a scientist, and that's my dad who encouraged me along the way. I like to show this because it shows that I was a good student. I got A's and B's, but also it points to the signature of one of my favorite teachers in the 4th grade and the 6th grade, Wally Vane. And also it allows you to see the marvelous signature of my dad. He wrote everything like that, including letters, checks. People would, he'd write a check to people and they would not cash it for a while because they wanted to show all their friends. So I was an amateur astronomer, took some pictures, and this is my first published scientific result. In Sky and Telescope magazine, 1970, we had visited Virginia to see the total eclipse of the sun, and I observed shadow bands, which are a phenomenon that sometimes is visible during totality or just before and after totality. And here's the thing right here. Victor Ambros found the bands visible for over 4 minutes before totality and almost 3 minutes afterward. That's in a real magazine, my name. And it says his station was near Chincoteague, Virginia. This is my station. I think that's the first time I felt like a real scientist right there. So my path, as you heard, started at MIT. Finished in 75. I was influenced a lot by people around me. Here we are throwing a piano off the roof of my dorm. That was the first of what became an annual piano drop at MIT that still happens today. That's me. I was actually influenced a lot by other students. So for example, the Struhl brothers were people who I got to know, and they were doing research in a lab, and I realized, wow, people can do research in labs, and they were so enthusiastic about biology that I got converted to biology. Ed Gruberg was a really important mentor of mine, taught me how to read literature and write a paper. I learned so much from David Baltimore, I can't possibly start to enumerate here today. Ditto, Bob Horvitz, although I'll try to mention a few things that Bob taught us. And then I had these faculty positions. I was recruited to UMass by Craig Mello, who used to be, before that, my PhD student. And so I have this Nobel connection that's, I think, fairly extraordinary. I've worked with 3 Nobel laureates. So I'm here to tell you this is an improbable life. So the story of this project begins in 1980, Bob Horvitz's lab. And shortly before then, and then soon after, Bob and John Solston put together different parts of the development of this worm into a complete cell lineage. And the way they did this was they sat in a room looking through a microscope, taking notes, watching cells divide in living animals, right, and drawing them as they divided and moved around, underwent programmed cell death, migrated here or there, and differentiated. And they did that enough so they were able to draw this lineage and say that it's invariant. They never, the animals in wild type never make a mistake. They do this lineage with this timing of cell divisions and these fates associated with these patterns, from animal to animal. So that was part of why the system was attractive to people, because it seems you could follow, analyze development on a cellular level in real time. I think also in retrospect, this invariance of the lineage offered to us a way of studying how developing animals are able to robustly develop given the stresses of everyday life. These animals live in the dirt. They have to eat whatever bugs they can find, bacteria. They live over a wide range of temperature. But the cell lineage development is invariant regardless. So the big question was, how is development encoded in the genome of an animal? Bob is using a genetic approach, which is the unbiased approach. You find a mutant, and then you follow that mutant and find out what it can teach you about the system. It's not based on presupposed ideas about how things work. So the model organism premise is pretty cool, right? The worm is a tiny person in disguise. And so that means if you study developmental mechanisms in one animal, it'll teach you about other animals. And the rationale for this is basically that we have a common ancestor. That's pardon my fanciful bilaterian ancestor, but the idea is that that animal had an endowment of gene regulatory network machinery that possessed latently everything for all the animals after that. And we now know from genome sequences, estimates vary, about 5,000 genes that are completely conserved. And it's that genetic toolkit and how it's adapted and how regulatory interactions and mechanisms are brought into play are underlying the diversity and complexity of animals. So the genetic approach is you've got a mutant and you've got a phenotype, right? So you figure there must be a mutation, and there must be a gene there. And you can verify that, as you all know, by Mendelian crosses. Okay, so there's a gene that's going to be in the DNA, so you got to find the DNA, figure out a gene product, learn what you can about potential biochemical activity, and ultimately you can relate that to your phenotype and how the mutation changes the gene, and make some inferences about normal function of the gene product, right? And in the 80s, this was a lot of work, right? Starting from a mutation and a phenotype, it could take easily 5 to 6 person-years to even, as we say, clone the gene so that you have the DNA and the sequence. So how do you choose what mutants to follow? And Bob recommended that we should follow these developmental timing mutants. And they're very interesting. You've heard about them. One is this LIN4, loss of function mutant, which I'll tell you about first. The 0 there means the mutation is, we think, knocking out the activity of that gene. And it turns out the mutation we worked with was indeed knocking out the activity of LIN4. So that's what happens to the animal when it doesn't have LIN4. It ends up developing in a pretty crazy way. Many of the cell lineages in the animal are repeating what normally would happen only in the first larval stage. So you can see, the blue circle and the pink circle, those are different patterns of cell division and cell fate. The blue thing is basically a neuroblast is produced there. And in the LIN4 mutant, things that happen later don't happen at all, and throughout numerous lineages, you see this happening. And this was worked out by Marty Chalfie, working with Bob and John Sulston. So when I came to the lab, this was known. It was a really interesting mutant, LIN4. And it had, so Bob taught us to make simple models, right? Okay, the phenotype is you repeat the first larval stage, you don't make the second larval stage fate, so it must be that the normal function of LIN4 is to specify the second larval fates and or prevent first larval stage fates. So when you remove the gene, look what happens. You repeat the first larval stage phase. These we called heterochronic mutants as a nod to the concept of heterochrony in evolution, where, for example, ancestral juvenile traits are maintained in a descendant generation. And so, this mutant is basically like that. It's retaining ancestor juvenile traits. So the opposite kind of phenotype, this was actually identified by Chip Ferguson in Bob's lab. These mutants, the LIN14 loss of function, do not do the fates that normally would happen in the first larval stage, and they skip right to the second larval stage. And this happens in many different cell lineages, so it's a sort of a global kind of transformation in cell fate to a later cell fate. So again, our simple model is that LIN14 could specify L1 fates and or inhibit L2 fates. So these two opposite phenotypes leave us with two nice little models, right? And Bob taught us to make regulatory pathway models. And so there's two alternatives, right? Lin14 might inhibit Lin4, which is specifying L2 phase. Or Lin4 might inhibit Lin14, you know, and that will control transition from L2 to L3 phase. So which is it? And so, all right, get ready. This is the simplest experiment in the world, right? You make a double mutant, okay? And what happens? A Lin4, Lin14 double mutant looks just like a Lin14 null, single mutant. So Lin4 doesn't matter if Lin14 is missing. So the idea is that's the pathway that must be the case. And so, you know, considering this, it's interesting to contemplate that, okay, what's going on in these mutants is cells are being transformed sort of in their cell fate in a temporal direction. And this implied that in these lineages in the wild type, that cells were being produced, stem cells essentially were being produced with similar developmental potentials. So the cell in this lineage in the first larval stage has both potentials for doing that L1 developmental program or that L2. Ditto for that stem cell produced at a later stage in that lineage. And what happens in the mutants is the fates get transformed such that what normally gets whatever option gets normally chosen at that stage is not chosen properly. And according to our nice little model at the bottom, that would be accomplished by Lin14 being present and then absent under the repression of Lin4. So this was interesting and justified putting a lot of effort into maybe cloning the Lin14 and Lin4 genes. And Bob recommended that Gary and I take this project with us to our respective labs, and sort of informally we split it up. My lab would concentrate on Lin4 and Gary's would work up Lin14. And then we were starting in each case with mutants, okay? I wanted to remind you of that. In our case, one mutation, Lin4, E912. You'll get used to that little number in a bit. And Gary's going to tell you about some really fascinating Lin14 gain-of-function mutations as well. All right, so we're getting started. What do we know? We have a genetic map, okay, recombination map. Lin4 is between blister 2 and dumpy 10. Our mutation is E912. This one, by the way, had an interesting provenance. This mutation was generated in the Brenner lab by feeding worms P32 labeled bacteria. And when they ate the radioactive bacteria, the P32 would degenerate and break their genomes and make mutants, and somebody screened through those populations of animals that were eating P32 bacteria for mutants, and E912 came out. So when we started this project, the whole project was based on a mutation that happened to arise by accident in the Brenner lab as a result of a P32 molecule, atom degenerating and shooting out whatever it does, a gamma ray that broke the DNA, okay? So that's how careers start. We were incredibly supported by the Medical Research Council MRC project led by John Solson and Alan Coulson. And about the time we started, here's 1988, this is a report in the Worm Breeders Gazette. I don't know how many of you have been around long enough to have subscribed to the Worm Breeders Gazette, but I happen to know there's a few people in this room who have. This was an amazing thing where it was, you know, before the internet, basically, people would post their unpublished results. And Alan and John would periodically post the latest physical map. They were working out the overlaps between all the clones that they had collected, DNA clones. They would share clones with us, of course, send us advice. I found a couple of letters from Alan and John. And this is, Rosalind worked with this specific map right here. You can see each line is a clone piece of DNA. Cosmids or yeast artificial chromosomes, the longer ones, they had worked out these overlaps by a high throughput fingerprinting method. But they didn't know where the genes were, right? So they were working in parallel then with labs like ours and dozens of other labs to put the genetic map in register with the physical map. And of course, this was a prelude to what became the C. elegans genome sequence. So dumpy 10, for example, it's a collagen. Somebody assigned it there. Look, it's just written in hand. So now we know, okay, so Lin4 must be off to the left, right, somewhere. We don't know how far. So Rosalind's first task was to identify clones from these collections here that overlap strain-specific DNA variations, or RFLPs. Okay, so hang on to your hats. If you're a geneticist, you'll like this. Otherwise, whatever. These are beautiful things. We wanted to find a piece of DNA that we could have in our hand and that we could say this piece of DNA has the following phenotype. It recognizes a piece of the genome that differs between two strains. And we can assay that difference by a southern blot. You can look up southern blot, young folks, on Wikipedia if you want. And you can map it now. It's a genetic trait. So you make a heterozygote between one strain that doesn't have this polymorphism and another strain that does. It's RW7000 strain. It has lots and lots of transposon insertions across the genome. So this allows you to detect these polymorphisms pretty easy. And you take a probe and probe it to your southern blot of these recombinants. You pick a bunch of recombinants that are all dumping on LINs, and then collect those and analyze them by southern blotting and ask which ones retained the MAP12 trait or not. And that maps that particular piece of DNA to a position genetically near LIN4, right? And much closer than dumpy 10. That's Rosalind's toehold in the genome. So using the MRC, Alan and John overlapping clones, she marched along and basically tested them as she went for whether or not the clones could, on southern blot analysis, detect differences between the E912 mutation and wild type. So think about this for a minute. If E912 had been a point mutation, right, there's probably not a chance in the world, really, unless that point mutation affected a restriction site. All right, so now we're getting gory, and if you're a molecular biologist, you can appreciate that. So the chance of finding a LIN4 E912 mutation was risky, right? But she found it. And it turns out that, so for example, here's a blot with two different enzymes digesting DNA from LIN4 E912 versus wild type, and you can see the difference, right? The wild type is much brighter and it's different in size. That's because a lot of that piece of DNA that she's using as a clone is deleted. And so, by the way, for these experiments, Rosalind benefited a lot from some new technology. This is like late 80s hot technology called pulse field gel electrophoresis that enabled you to take these yeast artificial chromosome clones that Alan Coulson and John Sulston and Bob Waterston were producing and mapping, and you could purify them using this. And she used this. This is an MJ Research controller. This was the first product of what became MJ Research, which some of you may recognize as a major supplier of thermocyclers for a number of decades. And this was made by Mike Finney and his brother John. Mike Finney was a graduate student, I think, in Bob Horowitz's lab at the time. So Rosalind took up the task now of sequencing this piece of DNA that was detecting the E902 mutation. But here's a problem at this point. Okay, so it's a big deletion that allowed us to see the lesion, but it doesn't really tell us with much resolution where LIN4 is. So we needed a point mutation. And Rana took up the task to conduct a series of high throughput, high volume crosses where the males are mutagenized and then crossed to a strain that is E912, right? And it's marked with a dumpy so that you can tell self from cross. And so the cross progeny will mostly be normal because E912 and the dumpy are recessive. The goal is to find a rare animal that looks like LIN4 E912, not because it's homozygous for E912, but it's heterozygous for E912 plus a new allele that knocks out LIN4, right? And LIN4 seemed to be a small target, right? Because these were very rare, unexpectedly rare. But she got one, and it's a point mutation, and it's called MA161. So that told us that the LIN4 gene product must traverse that part of the sequence, right? What about the boundaries of LIN4? So the strategy that Rhonda and Rosalind used was injecting clone DNA into C. elegans to generate transgenic worms. This methodology was worked out, by a number of people, including Craig Mello optimized it. And it was magically, so to speak, you can just inject into the worm DNA, and at quite high frequency, the progeny will now have that DNA, and it's inheritable. So you can ask, does the DNA that I inject confer wild-type Lin4 activity, meaning will it rescue the phenotypes of those Lin4 E912 mutants? And that clone did, a smaller clone did, a smaller clone rescued, an even smaller clone, and finally, 700 base pairs clone rescued Lin4. So this is a really small gene, and that started to pique our interest because even as a protein gene, this is going to be a small protein. So we felt we were onto something interesting. So the Lin4 gene has to be in there. We want to find the transcripts. So those sequences are used to probe, as probes, try to find the transcripts through various hybridization strategies. And Rhonda went on maternity leave, I think, and so I did for a little while. I actually did some stuff, and doing these ribonucleotide protections. And here you have a radioactive probe. You hybridize the total RNA of wild type or your E912 mutant, and you ask what size of a piece of the probe gets protected from ribonuclease because it's formed double-stranded. And we got really excited because it's 55 nucleotides. So I'm telling you, wow, we knew we had something unusual. The gene is small, and the transcript is only 55 nucleotides, or maybe a little bit more than that, depending on interpreting the probes here. So we knew it was small. But then, so I presented these data at a floor meeting, a research in progress, and our friend Margaret Barron said, "What's that at the bottom of the gel?" And I said, schmutz, I don't know. And she says, well, maybe you should look at a shorter exposure. And sure enough, the major transcript, which is present in the wild type, present in the rescued strain, absent in E912, absent in the control, is really small. So the major transcript is small, about 22 nucleotides. And Rhonda now really nailed this down with northern blots and S1 nuclease protection and primer extension experiments. So there we were. Now we definitely had something unusual. There's the MA161 mutation in one of the nucleotides that's in that major transcript. The minor transcript, longer, folds into a hairpin. We now know that's the precursor that gets processed out by Dicer and captured by Argonaut. So, and there's MA161. So the thing is, we think, okay, this thing has a very small transcript. It's really unusual. And we're going to report to the world that that Lin4 does not encode a protein, right? And I think that I was annoying, perhaps, to Rhonda and Rosalind because I thought, okay, we got to prove it. We got to prove there's no protein whatsoever. I mean, there are some start codons, but of course there's some stop codons too. So Rhonda and Rosalind did some mutagenesis, inserting out of frame, substituting in a stop, all those constructs rescued, so those putative upstream orphs can't be real. And then they did evolutionary mutagenesis. Rosalind cloned Lin4 from 3 other nematode species. These species are maybe 20 million or so years different from C. elegans from each other. So you're letting evolution do the mutagenesis. And importantly, all those clones from the other species rescued C. elegans Lin4. So they have Lin4 function, but if you look through the sequences, you basically cannot find a conserved protein. So we nailed it. There's no protein produced from that gene. So in June 1992, Gary and I got together and compared sequences because we had the small RNA sequence. He had sequence from Lin14 3' UTR, and we matched that up. We got on the telephone. I don't know how many of you recognize that as a telephone. And it's a landline, and there was this complementarity between these 3' UTR elements in Lin14, and importantly, Gary's lab has shown that those elements were conserved in multiple nematode species, just like we had shown that the Lin4 was conserved in sequence. So the complementarity was conserved. So in that moment, we knew the mechanism. It's really wonderful, because it's rarely that you can kind of inspect data and know the mechanism. So that was definitely fun. So we published those papers. Nowadays, by the way, we know that Lin4 is a little bit shorter than what we had it. We're being generous, given the vagaries of the methods at the time. Okay, so what did we do then? So from 1993 to 2000, our lab was doing some heterochronic stuff. The pathway was a little bit crude in the beginning, about that time. We knew that there was Lin4, Lin14, there was Lin28 was involved, and these were controlling early larval fate transitions. The Lin29 was controlling later transitions, and Rugby cloned Lin29, showed that it was actually a conserved zinc finger protein. And Eric Moss, working with Rosalind, cloned Lin28. That was also a conserved protein, in this case an RNA binding protein apparently. Also, they found it was targeted by Lin4. So this Lin4 mechanism, whatever it is portable to another gene, as long as you have sites in the 3' UTR for Lin4. And then Rhonda did some nice experiments showing that Lin4, in fact, acts as a switch. Phil Olson did some interesting work on the mechanism of Lin4 repression of Lin14. I'll mention that in a few moments. And then working with the Ruvkun lab, found that Lin41 is a conserved protein and it's targeted by LET7. So there were 3 conserved proteins in the pathway. So the heterochronic pathway had, gained a lot of cachet at this point. But, up until 98, though, Lin4 was the only microRNA, and even at 99, Lin4 and LET7 were the only microRNAs, and they're only in C. elegans, right? So, but what was going on in parallel was a really interesting consilience of microRNA and RNAi phenomena. So Fire and Mello discovered RNAi and C. elegans, and Hamilton and Balcombe showed right about the same time that in plants, RNAi was associated with a blooming of small RNAs of about the same size as microRNAs. So it really looked like RNAi and microRNAs may be mechanistically related. This was really nailed down by the Mello lab working with Ruvkun, showing that Argonaute proteins are central to RNAi and microRNAs, right? So this really told us that, look, maybe microRNAs, Lin4 and LET7, are evolutionary adaptations of RNAi for gene regulation, right? And let me remind you, RNAi is generally, a viral defense mechanism where double-stranded RNA is detected. Foreign double-stranded RNA usually is detected. Viral replication intermediates, for example. Dicer cleaves that up into small RNAs. They go into AGO. One strand and it goes into AGO and it destroys the invading RNA. So microRNAs are double-stranded RNAs that are processed by Dicer, but the double-stranded RNA comes from the genome. It's in a gene that's evolved to regulate other genes, right? So microRNAi is ancient, so are microRNAs. LET7 is conserved across all the animals. This paper, when it came out in 2000, was a real watershed. And personally, I didn't even know these results. These guys had kept it secret, right, for a maximum drama. So, when I opened Nature and read this, I thought, holy cow, wow, So microRNAs, LET7 is ancient, derived from RNAi. The bilaterian ancestor had LET7. Look, it's been 400 million years. It's had plenty of time. It's just, life has to evolve other microRNAs besides LET7. And we just haven't found them, right? Because they didn't present themselves to us by mutation, right? Okay, so in a model organism. So Rosalind and I got excited, and we taught ourselves all kinds of new stuff, like making cDNA libraries from small RNAs, growing up these, making libraries. We just hadn't done this stuff before. We found finding somebody who could sequence little short inserts without charging a fortune, or as the Whitehead wanted to do, be co-authors and charge a fortune. So then we could match them to the genome, and sure enough, the genome where they matched could be folded into a hairpin, and so they look just like Lin4 and LET7, but they're different sequences, and many of them are conserved in other organisms, including flies and humans, just by sequence match. So we thought, well, we got to write this up. It's going to be cool, But then one day I get an email from the editor of Science and says, can you review this paper? Here's the abstract. And the abstract from the Tushel lab. And it says, we've cloned many new microRNAs, from humans, and they matched Drosophila and C. elegans and stuff. And so there's way more microRNAs out there than Lin4 and LET7. So it's like exactly what we wanted to say. So I said, no, I can't review it because we have similar results. And is it okay if we send our manuscript to be, considered at the same time? And he said very kindly, he said, yes, but you get it to us by the end of the week. And there's another paper from the Bartel lab, So I'm telling you, by the skin of our teeth, we got this paper co-submitted and co-published side by side with these other papers. And together, the scope of these papers varies, and ours was focused on C. elegans, but basically the message from these three is there's lots of microRNAs, new ones, and a lot of them are conserved, like LET7 in all these animals, in flies, worms, and humans. So lots of other papers followed from that and sort of filled things in, and including showing that Lemire 100 is in Nigeria, meaning that it's even more ancient than the bilaterian microRNAs. And nowadays we know of about 25 bilaterian seed families. And the way we refer to them as seed families is because position 2 through 8 of the microRNA, which is the part that nucleates binding to targets, is conserved. That's the part that gets fixed in evolution once a microRNA evolves. So, and by the way, there's other microRNAs in sponges, fungi, and plants, but they're probably evolved independently from RNAi. So there's maybe, 500-600 genes, microRNAs in humans, we now know, 100 or more. Seed families, a couple hundred, 100 to a couple hundred. How do microRNAs recognize their targets? Here's an early study that really kind of told us. This is a Brennecke paper showing that for these microRNAs and these targets, you can see base pairing predicted between the seed, and in some cases in the middle, seed only, and in other cases, seed plus 3' sequences. What's shown is, I think, 5 or 6 species of insect. So this conservation of the base pairing or the complementarity is used pretty routinely to apply confidence to the fact to the idea that these interactions are functionally real. So we search for potential targets of a microRNA computationally using the seed sequence, essentially. Now, how do microRNAs repress the target? So the key thing here is you notice that the microRNAs in animals don't perfectly match their target like siRNAs do in RNAi. That activates the slicer activity of Argonaut, and the RNA gets destroyed. It appears that evolution has contrived to allow animals flexibility in how they use their microRNAs. Most of the targets of all the microRNAs, essentially, are mismatched at least position 9 and 10 of the microRNA. So this gives the situation some flexibility. There's no slicing, no rapid destruction of the mRNA, and this allows what's called a programmable repression by other means. And we know that Argonaut recruits a variety of proteins, assembles complexes that apparently have specialized or programmable outputs to either repress translation in some fashion or to cause the mRNA to be destabilized, often through deadenylation, and then decapping, and then destruction. So there's lots of examples, and I want to emphasize, of microRNAs repressing targets without destabilizing the mRNA. So this is an early example from Brennecke et al. 2003, which in my opinion is actually still the best paper in the field of microRNAs. It's really amazing. Done by 2 graduate students in about 13 months, and published it in Cell. The Bantam microRNA represses HID protein production, and that regulates, is involved in apoptosis of the fly. So you can see here, these are wing primordia that they dissected out of the caterpillar. And the left panel is the protein detected by an antibody, and then the right panel is a primordia where they've detected the mRNA in this fashion. And you'll notice if you're a fly geneticist, they used this beautiful and elegant style of experimentation where they expressed HID in a stripe down the middle of the primordium, right? These are in flies that are not making the bantam, not producing the bantam. In flies producing bantam in this wing disc, you see the protein is essentially erased, but the mRNA is still there. So somehow the Hid-Bantam interaction ends up in translational repression of the target. Here's another example. This is familiar Lin4, Lin14. Phil Olson in the lab did these studies. If you take L1 larvae and grind them up and run them on a sucrose gradient, you can stretch out their ribosome-containing complexes, and down near the, as you go to the right on this graph, these are mRNA complexes with more and more ribosomes on them. The Lin14 mRNA, detected here by northern blot, you'll notice lots of it is associated with polyribosomes. This is in the L1 when Lin4 is not present, lots of Lin14 proteins being made. In the L2, when very little Lin14 protein is being made, surprisingly the Lin4 message is still associated with polyribosomes. So this suggests some kind of mechanism where the translational repression is occurring in association with polyribosomes, perhaps freezing them in some fashion. These conclusions were supported by the Andy Fires lab working with 21st century technology. So this partial seed match, the seed match configuration with some 3' pairing allows you to search computationally for targets. And each microRNA tends to come up, prove to be predicted to regulate multiple targets. And so here's where I want to make a really cool point, not a cool point, but an important point that we don't really know the answer to. Okay, this target scan program is a conservative one. It identifies targets without a lot of false positives. But what's fascinating is that, for example, C. elegans Let7 has about 134 predicted targets, but actually the Ruvkun lab showed by genetics that the vast majority, or most of the Let7 loss of function phenotype is due to de-repression of one protein, Lin41. So one of the interesting open questions in this field is, for microRNAs, like, what's with all the targets? So I'll just quickly do this. In this study with Chen and Rajewski did was to basically, let's compare 3 species, flies, worms, and humans, for how many microRNAs they have in common. There's about 20 conserved seed families, and for about the 5,000 genes that are conserved across these 3 organisms, there's a very small minority of those genes that happen to be targeted by the same microRNA in all 3 species. So there's quite substantial evolutionary fluidity in targeting, and that's thought to reflect perhaps an adaptive role for gain and loss of microRNA targets. Now, at the same time, there are deeply conserved targets of microRNAs. And one kind is this Let7 Lin28 little epigenetic circuit where the RNA binding protein Lin28 directly regulates Let7 and Let7 directly regulates Lin28. And this controls pluripotency versus differentiation in many, many contexts, even in the same animal. So it's, in C. elegans, it's employed in our heterochronic pathway in this fashion. So one mode of action of microRNAs is developmental switches, like Lin4 and Lin4 on Lin14, Let7 on Lin28 or Lin41. Another interesting mode is related to switches. It's in this example, these neuronal microRNAs can be sufficient to reprogram directly fibroblasts into neurons. Another mode of action of microRNAs is coordinating suites of genes. And in this case, miR1 is regulating multiple components of a multi-protein complex. And this turns out to be important for muscle physiology. Another role that's been shown in a number of contexts is microRNAs are involved in gene regulatory networks where they basically buffer the network output from noisy input. And noisy input is an inevitable sort of trait, let's say, of transcriptional regulation, for example. Okay, one of the ways I love these experiments where people ask, well, what do microRNAs do? So let's just make a fish without any microRNAs. And I don't want to go into detail, but it's a beautiful experiment where Antonio Giraldez, working with Alex Shear, made these fish that have no microRNA. What happens? They make a fish, okay? So you don't need microRNAs to make a fish. But the fish are simpler than they normally would be. They lack a lot of cell types and a lot of morphological features that normal fish have. They're also very stress sensitive. So, the take home is microRNAs are good for promoting developmental cell complexity, and conferring physiological and developmental robustness. Here's another example from flies. Let's make some flies without microRNAs. And so the Carthew lab did this. Not surprising, an AGO1 mutant, the microRNA Argonaut, those mutants fail in development. That's if they're developing normally. If you contrive to slow their metabolism by removing insulin signaling so they develop very, very slow, at very, very slow metabolic rate, they can make flies. And similar results with if you're an aficionado in clones in the eye. So the idea is that they propose a really, I think, an interesting idea that microRNAs enabled rapid high energy growth and development, which would be an obvious adaptively advantageous innovation. All right, so they promote organismal complexity and stress-resilient living. Now, of course, stress resilience is intricately related to disease, right? And so this brings up the subject of, okay, what do microRNAs do in human disease? And I don't have any time here to cover the vast literature in this area. I just want to tell you one story that has been important to us because it's enabled us to get involved in our lab in a way of shifting from purely C. elegans, developmental genetics to a kind of partially C. elegans developmental genetics, but with some human context involved. And so I want to tell you about these human Argonaut mutations in neurodevelopmental disorder. And so these are mutations that occur spontaneously in people. They're not present in the mom or the dad, but the child has a neurodevelopmental disorder, delayed. It's many, many different kind of let's say indications, varies from child to child and so forth. These were striking because these are deletions of multiple Argonaut genes. So Argonaut 1, 3, and 4, AGO1, 3, 4 in humans are clustered together. These are microRNA Argonauts. There's one more microRNA Argonaut, AGO2 elsewhere. These mutations are knocking out multiple Argonauts, but they never found individuals that carried a knockout of a single Argonaut gene. Okay, so that suggested that to this indication, neurodevelopmental disorder, required that multiple Argonauts be compromised somehow. Okay, but there's the most prevalent mutations are point mutations, but these are all single amino acid changes in, as it turns out, evolutionarily conserved amino acids in AGO1 or AGO2, and now there's AGO3 mutations have arisen in some individuals, or been found in some individuals. So here's 4 examples of the mutations that come up in these patients. All right, if single amino acid change can cause this condition, and I've just suggested that broad disruptions of multiple genes are required in order to get the condition, maybe could a single amino acids variant actually broadly disrupt microRNA regulation? And I'm going to show you why we think that could be the case. Because we had, Anna Zinovieva in the lab, had happened to accidentally come upon some C. elegans Argonaut mutations that also were like that. And one of them was right next door. They were like that in the sense that the mutation had broader effects on microRNAs than would have had if the gene was deleted. How does this work? In wild type, there's 2 Argonauts for microRNAs. In C. elegans, they're redundant. So if you knock out one of them, actually the remaining ALDS2 is almost sufficient to take care of the job. However, if it needs a so-called antimorphic mutations, the mutant protein is present, expressed, and runs rogue, let's say, right? So it associates with microRNAs, but it can't repress targets properly, and so it sequesters microRNAs away from ALDS2, and so it kind of, the dysfunctional Argonaut poisons the normal redundancy. So could the Argonaut syndrome point mutations poison the redundancy of other human Argonauts? So we decided we would test that in C. elegans by modeling it. I know I'm running out of time, so if anybody's worried about that, I will accelerate, and I'll be done. So we modeled them in C. elegans, and this is, CRISPR-Cas gene editing is so amazing and easy in C. elegans, so we were able to make these mutants and look at them. And two of them had strong developmental phenotypes that we could show were stronger than the null. So this allowed us to suggest perhaps, indeed, the point mutations in humans were causing this condition because they were disabling or poisoning, let's say, the normal redundancy of microRNA argonauts. Okay, the other ones are milder, and I won't say much about them. And so we then wanted to do molecular phenotyping. What do we want to know? We want to know what's the microRNAs that are expressed and associated with these proteins. And also want to know what the gene activity impact is. And we use ribosome profiling, which allows you to quantitatively measure and identify across the genome all the sequences that are associated with ribosomes. And so for each gene, you can get a measure of how active those mRNAs are on ribosomes. At the same time, you can quantify the total amount of each message. And that gives you, for each message, thousands of which you're measuring all at once, a translational efficiency. So what do we basically find? And this is where I'm going to finish up. We basically found that the different mutations in C. elegans, modeled in C. elegans, can have distinct effects on the microRNA functionality in that animal. Some microRNAs can be affected in one mutant and not in others. Also, the downstream disruptions of gene expression can be quite distinct with these two mutations. And so that also allowed us to take the liberty of considering that maybe the manifestation of these mutations in patients being so different amongst patients between different alleles could in part be due to the differences in the downstream protein balance or imbalance that's being produced. And one of the most amazing things about this is that we found that focusing on targets of microRNAs that are disrupted in both mutants, that those targets are disrupted differently. Primarily in the H751L, those targets are disrupted on the level of translational efficiency and not mRNA abundance, vice versa for the other one. So we think that this offers a basic sciences opportunity to investigate, basically by using genetic approaches and now combining with biochemical and structural analysis, which is going on in other laboratories, we can make some really interesting inferences about mechanism. Now, so there's also, however, the question of what can these studies in C. elegans tell us about the human condition? And I think it's not much in terms of particulars. That is because, remember, humans have many more microRNAs than C. elegans, and I've already showed you how the target repertoire of even the conserved microRNAs can be very different across species. So we don't think it's the particulars of the gene expression disruptions that are downstream that we observe in C. elegans that could be, informative about the human situation, but it's more the character of them and the differences between mutations that we can resolve in the experimental system. And this is my last slide. Argonauts syndrome is a very, human phenomenon. It involves patients, families, clinicians, support teams, and so forth. And it's the patients that are gifting to us the identity of their mutations that allows us to do experiments, genetic or otherwise, and make inferences about fundamental mechanisms that are going to apply to how argonauts and microRNAs work in all sorts of contexts. But at the same time, of course, we're hopeful and optimistic that we can learn enough from these studies to be able to inform the disease. So in my last 15 seconds, I'm saying thank you to you all for being here. Rosalind Lee and Rhonda Feinbaum for discovering the Lin4. For Gary, for sharing everything we've shared over the years. But most of all, to Candy Lee for everything. Thank you.
I now have the great pleasure to introduce this year's Nobel laureate in physiology or medicine, Dr. Gary Ruvkun, our second star. Gary Ruvkun was born in Berkeley, California. In 1973, he received a Bachelor of Arts with a major in biophysics from the University of California, Berkeley. In 1982, he received his PhD from Harvard University after studying bacterial nitrogen fixation genes in the laboratory of Frederick Ausubel. Dr. Ruvkun then started his postdoctoral research jointly between the laboratories of Walter Gilbert of Harvard University and Robert Horvitz at the Massachusetts Institute of Technology, remaining there until 1985. He then became an assistant professor at Harvard University and started his own laboratory at Massachusetts General Hospital. Gary Ruvkun is now at Massachusetts General Hospital and professor of genetics at Harvard Medical School in Boston, Massachusetts, USA. Dr. Ruvkun, we are very much looking forward to your Nobel lecture entitled A Vast and Ancient Hidden World of microRNAs Across the Eukaryotes. Please join me in welcoming Dr. Ruvkun on to the stage.
Thank you. Thank you very much. Am I on? Yes, I thought I might have unplugged. It's really wonderful to get this kind of reception. It's been a wonderful week, and the phone call from Stockholm was lovely. Thank you. So I'm going to talk about, our work on the vast hidden world of microRNAs, and it's many of my students and postdocs over many years. And I just want to remind you for a second that what we're talking about is watching these lineages in wild type. They make a transition, and in a Lin14 dominant mutant that I'm going to be describing, or the Lin4 microRNA mutant, you suddenly fail to make this switch, and you keep going L1, 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 L1. Whereas if you don't have Lin14 protein, you never get to L1 and you go straight to L2. And this was the sort of the worm's blessing and curse, was to use lineages as a way to assess what a cell is wanting to be and trying to be. So these patterns of division tells you what the cell thinks it is in terms of time or space. But as developmental biologists, talking about lineage was very strange to the developmental biology community. And so we would go and present these sorts of lineage mutations, and at developmental biology meetings, all the other people who were working on cell-cell interactions would think we're very much interested in ancestry and who your parents are, and developmental biology was all who your neighbors are. So worms were sort of an outlier in biology, but we kind of liked it. And that's that the worm community was always a little bit separate. And so the microRNA story is a bit of that, where we sort of think different, right? And it works to think different sometimes. And so the gain-of-function mutant was really interesting to study, and using the same kind of chromosome walking that Victor described, we got to the Lin14 gene. And the interesting part of it was not the protein coding segment, which is, again, a little bit strange. Usually what people are interested in is the protein. It's homologous to something that's related to a disease. And, and proteins are sort of have a lot more letters to them, right? There's 20 amino acids, and if it matches, it's significant, and DNA is harder to match. And these gain-of-function mutations turned out to be in the 3' untranslated region. So this is downstream. It's a 3.5 kb message. and about half of it is 3' untranslated. And there were 2 different activating mutations, and so that gave us a statistics of 2, which is way better than 1. And one of them was a 600 base deletion that activates the gene, and the other was a translocation that removes this back end of the gene. And if we look at the protein product, so we made an antibody, and this is the work of Bruce Whiteman and Il-Ho Ha, making an antibody to Lin14. It turned out to be a nuclear protein, and at the larval stage 1, it was abundant, and at larval stage 2, it was unabundant. And so it sort of fit with what the genetics was telling us, that the gene is active at the L1 stage and inactive at the L2 stage. And then the gain-of-function mutant failed to turn itself off, and it continued to have this nuclear protein at the L2 stage. And this nuclear protein didn't have any homology until a year or 2 ago, when AlphaFold was used to say that Lin14 folds in the same way as a couple of different things that don't have amino acid homology, but AlphaFold has saved our butts on having some homology, so we're going to the lecture tomorrow. Thank you, AlphaFold. And we can see this at a Western blot. Now, since Victor mentioned Southerns, it's time for an educational moment. A Southern blot is named after Ed Southern. Do you think a Western blot is named after Fred Western? No, it was a joke, right? A stupid molecular biology joke. And Northern blots aren't named after aren't we funny? Yeah, I know. There's a lot of room to be better than that. So here's the Western blot showing the protein going down by a factor of 50 from L1 stage to L2 stage, but in a Lin4 microRNA mutant, we didn't know it was a microRNA when we started, it doesn't go down nearly as much, so it fails in that downregulation. And again, this is Bruce and Il-Ho did this experiment. Now, to prove that it was a 3' UTR, this is in the days before GFP, right? 1993, GFP is discovered, 97? You guys probably know. But there was no GFP, but LAC-Z fusions were the coolest thing then. So you could take beta-galactosidase from E. coli and put it in front of the Lin14 3' UTR. So this is an animal that carries a collagen promoter, LAC-Z, the Lin14 UTR. It gets turned off. Oh, I'm sorry, that should say L4. Oops. It gets turned off at a late stage, but in a Lin4 mutant, it fails to get turned off. Or if we switch the UTR to have a non-temporally regulated muscle 3' UTR, it fails to get turned off. So the Lin14 UTR has something that turns it off and is responsive to the Lin4 mutant. And this is the paper that was back-to-back with the Lin4 microRNA paper. And so here's the Lin14 stem loop. This is the Lin4 stem loop, and this is the microRNA itself. And that Victor's lab showed is upregulated at the L2. And the way that upregulation works is there's these 7 sites. You can see where they are relative to the Lin14 gain-of-function mutants. So this one is a weaker gain-of-function than this gain-of-function that gets rid of all of them. Here's site 1 that has Lin4 on the bottom, Lin14 on the top, and it's Watson-Crick base pairing with this loop, 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 and that keeps it from getting cleaved. So here's the model that emerges, that Lin4 microRNA gets upregulated and that downregulates the Lin14 protein, the translation of the Lin14 protein. And this, again, it was done by Bruce. Here he is today. and Ilho. So that's where we were in the pathway, but then LET7 emerged as having heterochromic defects as well, and so now I'm going to tell you the story of LET7 and Lin41 that happened a few years later. And so the LET7 phenotype is affecting a transition that takes place later, and what we're watching is the pattern of divisions of these cells along the lateral hypodermis of the animal, and these are cells that have different temporal patterns, and the last pattern is to not divide and to form these sort of racing stripes on the animal. And the wild type has a perfect racing stripe, and the LET7 mutant fails to develop those racing stripes because it keeps going L4, L4, L4. It never gets to adulthood, which some of us can understand very well. And so getting to the LET7 gene was the work of Brenda and Frank, and this was in collaboration with Bob Horvitz and Michael Basson, who did much of the genetics on LET7. And so they had to walk into the region and figure out that the gene corresponded to a microRNA, and we actually didn't expect it to be a microRNA, even though there was another microRNA in the Lin4 had already been discovered 5 years earlier. But we were imagining it was going to be the regulatory cascade, another set of transcription factors or something. But it turned out that it was a point mutation, and it was the point mutation helped us guide us because it said, well, that's the active site of whatever it is we're working on. And so when they did a northern blot, again, funny joke, northern, not southern, this microRNA is 22 long. It's not expressed at early stages, and then suddenly at the L4 stage, when we know it functions from genetics, it gets turned up. This is a lethal mutant, and so you can then do a genetic screen where now how do you work with a lethal mutant? Well, it's temperature sensitive lethal. So it's lethal at 25 degrees, but if you grow it at 15 degrees, which is fine for a worm, you can grow it up, do a mutagenesis, wait 2 generations, which is the glorious thing about a worm is you're talking a week. It's not 6 months to get 2 generations, it's a week. Or humans, it would be, 100 years. A week is better than 100 years. So you could ask for suppressors out, and out of that, sorry, where am I? Here we are. This is the work of Brenda and Frank, and they're in the audience too. And out of that, the suppressor turned out to be the target gene Lin41. So Lin41 is a conserved protein. It has lots of paralogs, both in the worm. These are all the worm genes, but these are the mammalian homologs, and it's a ring finger protein, probably a translational control factor. And when we looked in the 3' UTR of Lin41, we saw conserved complementary. So here's the Lin41 sequence going that way. This is the LET7. This is one LET7 microRNA, second LET7 microRNA. Similarly bulgy, not the same kind of bulge. This is more of sort of a 2-base bulge instead of a 5-base bulge in the case of Lin4. We don't know why that is. And so we have now a case of a microRNA here, the LET7 here, and in fact, there's paralogs of LET7 that I won't talk about that do these transitions here. And you can see in the lineage of Sulston that there's these waves of division. So here, there's one wave of generation of cells here, a second wave of generations here, a third wave, a fourth wave. And those are all the in-between molts. So at this point where there's no divisions is when the animal sheds its cuticle. And this idea of having molting is not unique to C. elegans. There's a whole large clade of animals called the ecdysozoans because they do what's called ecdysis, which is to shed your cuticle. And that's how insects, you go from larval stage to the larval stage as well. So there's something about microRNAs and larval staging for sure that comes out of C. elegans. And so then we could look with the LET7 sequence and look for homologs. But here we're aided by the emerging genome sequences in 2001. And so at that moment, there's a human genome that's available. And in fact, our hero, John Sulston, who did all this lineage work, so he did this sort of working almost alone, maybe 2 or 3 people, including Bob Horvitz, working on this, but it wasn't a giant team. And then he aids us, as Victor said, in sort of chromosome walking. And I have to tell you that I was important in this because Sulston came and saw me doing chromosome walking and said, what a waste of a brain. That you're a pretty smart guy and it's so boring. I could walk from 100 places. You're walking from one place, I'll do it from 100. And so that's why Victor said we would communicate with them and they'll help us, and they did it with everybody, putting together the Genome Project as a gift to the field. And there's such a thing as thinking with your hands. And so he realized how genomics worked with he and Alan Coulson, basically doing cosmet preps and fingerprinting, a very, labor-intensive stuff. He's not sitting in a room thinking. He's thinking with his hands. And so at that point, he ramps up to do the Genome Project, and he goes from 2 or 3 people to 100 people. And in fact, he's a third of the human genome, doing a third of the human genome. And so when we wanted to look for a homolog, we could just take this very short DNA sequence and do it was before BLAST, but you would just say, well, is there anything that looks like this in a third of the human genome? And that was done with a modem from home. So I'm sitting at my desk and you get the data and it comes back and it says, yeah, there's a perfect homolog in C. briggsii, of course. Well, that's not a big reach, but zebrafish, human. At this point, all there was human, right? And zebrafish and all these other chimps, et cetera, they show up in the next 5 years, right? And now there's 20,000 eukaryotic genomes. There's another 500,000 bacterial genomes. I can't begin to, you all maybe know how wonderful it is to be a molecular biologist today, right? That you have all this data that you can just look at and like, oh, I think I'll blast this. It's all I do. So there's Amy, and of course, guess what that is? That's the LET7 sequence. And she's now and she lives in California, so you can, that's how people are in California. I'm from California, I know. And here's Brenda again, working on this project as well. And both of them are here today. And then how about proving it, right? So that was a database search, modeming into a third of the human genome. So part of this project was me being charming and begging for RNAs from people. And so Mitzi, who's also in the audience, sent us fly RNA, staged fly RNA, and Amy did northern blots. I won't belabor the joke. And you can see that there's nothing early, and then it comes on at sort of the equivalent of the L4 stage of a worm, if you don't know too much biology. And so you can see that this is a late stage regulatory event. It doesn't prove that this does anything. It just says that it's regulated temporally in the same way. And in the Lin4 case, it was not, you don't find a homolog in the fly. We could also do it in zebrafish with RNA that Mark Fishman sent. And here's LET7, not there in the early fish, but sort of later in the development of a vertebrate, right? So that's getting closer to a human. It's still kind of it's a fish. but it's temporally regulated, and it sort of validates what the human genome comparison said. It's just, but it adds to it that it's temporally regulated. And then we can look in human tissues, and you can see that it's in the brain or kidney, liver, et cetera, and sort of less in cells that are more totipotent or dividing and not so differentiated. But it for surely, what it did say is that it was expressed as a 22 nucleotide RNA as well. So summarizing here all the organisms we looked at, and again, people sent us, RNAs from just the weirdest organisms. Super fun. Some of them came from Australia. So plus means we could see a band. We didn't get developmental series from everybody, but almost every place we looked that's in the bilaterians, and these are the ecdysozoans, but almost all of them have it, and almost all of them are developmentally regulated. And we did not detect LET7 in these more basal eukaryotes, but other microRNAs have been detected in these, and so LET7 didn't happen to be conserved. So this told us that microRNAs were going to be in many different organisms of the eukaryotes. And then in the end, we have sort of hundreds in the vertebrates, almost a couple hundred in the invertebrates, a few in sponges. There are no microRNAs in the yeast, but plants have also hundreds, and none of the plant microRNAs are equivalent to the animal microRNAs. So what it tells you is that the common ancestor of animals and plants had microRNAs, and I'll tell you about Argonaut in a minute. They use the same Pee-wee protein, the Argonaut, to present those microRNAs, but they've evolved different microRNAs, and it's either because evolution is too fast, but it's really only a factor of 2 between plants and animals. And so it might tell you that there's something different, and what I think, and I'll probably get to it, is that the time scales for how it works in a plant versus an animal is very different. We have microsecond things going on in our nervous systems, and maybe I'm just an animal-centric view of the universe. Maybe plants don't have millisecond type things going on. Their sort of temporal sequence of responses is slightly more slow, and that might be what's different, and I'll get to that in a bit. So what are some other conserved microRNAs? It's not just LET7. Another really highly conserved one is called MIR1, most likely because it was the first one. I can't remember who discovered MIR1. Probably Victor. Oh, there it is, yes. Sorry, Victor. It's hard to keep track. So this is really highly conserved. And I bet it wasn't the first one you discovered. You just put it top of your list. And actually Victor's lab shows that it's involved in muscle development, and it does something in our heart muscle in a mouse. And we did some work on it as well. One of the ways to study these microRNAs is to take the gene for it. And even though this is coding for a tiny RNA that folds back, et cetera, you can still slap GFP onto it. We had evolved by 2001 from LAC-Z to GFP because Marty Chalfie had made GFP what everybody uses. And you can see now that this is expressed in body wall muscle and the muscle of the pharynx of the worm. So it's sort of a muscle-specific microRNA. And in fact, if you look at who the targets might be, you can see good conserved base pairing, that the MIR1 is conserved, as is the targeting of a acetylcholine receptor called UNC29. UNC is used to refer to worms that don't move. They act uncoordinated. And in this case, the uncoordinated is due to a muscle defect, a response to a motor neurotransmitter. And these cholinergic acetylcholine receptors are multiple transmembrane domains with multiple domains, multiple protein domains that heterodimerize. And in fact, in collaboration with Josh Kaplan's lab, we can see that the UNC29 protein is expressed at a lower level in wild type, but when you get rid of this microRNA, it now is expressed at a much higher level. And what was strange about this is that we then expected the microRNA to be, microRNA mutant to make more of that protein, to therefore be more resistant to levamisole, and we got the opposite result out of that. We don't know why that is. One view is that there's other acetylcholine receptors that are also repressed, and there might be a relative mixture of the two that leads to the levamisole sensitivity. We also looked for more microRNAs. John Kim did this in 2004 in collaboration with Ken Kosik's lab with Anna Kraszewski, pulling out microRNAs from cortex. And we got miR-124 as one of the most abundant things that came out of that. And there again, we could fuse in GFP. This is very highly conserved between mouse and worms. And with a GFP, we could see that it's also neuronal specific. And in fact, miR-124 in a mouse is in the cortex and cerebellum, very particular to the brain. And when we looked at where is that microRNA on a polysome prep, so this is where monosomes are here, polysomes are where translation is really cranking out the product because multiple ribosomes have glommed onto the RNA and are gliding along, producing protein, multiple copies of a protein from a single mRNA. We can see that the microRNA is actually in the polysome fraction on a sucrose gradient, saying that maybe it inhibits translational elongation and not translational initiation, which is something I'm really interested in today. So I wanted to do 2 minutes on plant micros because they've been really interesting. So at the same time that the Bartel lab and the Ambros lab and the Tuschl lab were cloning microRNAs, this is a Bartel paper, and notice Brenda Reinhardt's on this one too. So Brenda's kind of the star of the show here. She was involved in this plant micro work, and one of them, called miR-156 from a plant, is very highly conserved in plants, all the way from Arabidopsis to rice. And rice is a long way from Arabidopsis. It's monocot versus dicot. These are like as far apart as you can be on plant phylogeny, and it's still conserved. And if you look at where it is, some of the complementary sites to the microRNA are in the 3' UTR. That's the light box here. But a bunch of them are actually in the protein coding region of the plant. And it's all perfectly conserved, and the base pairing is perfect base pairing. It's not the base pairing that we see with the loopy, bulgy base pairing in animals. And that's a blessing in terms of finding targets, because 4 to the 22nd power is a lot more statistically significant than 4 to the 15th power. And so it's easy to find targets. And if you look at who the targets are, there are a transcription factor called SBP, and this is the coding region of it, and that's what it codes for. And so you can see that the same thing is coded. Here are the ones that are in the 3' UTR. It's again, probably that's driven by a selection to target this coding region. And what's interesting about the plant micros is that they tend to work in a much more RNAi-like fashion of leading to degradation. So again, at the time that this micro explosion was happening was the explosion of RNAi. And sort of a key event was Craig Mellow's discovery that there's a protein that it's called a Pee-wee protein that mediates the presentation of these 22 nucleotide siRNAs in the RNAi pathway. And so the question was, do microRNAs use anything similar? And it's almost numerological reasoning, right? Because we're saying, well, this is 22 long, and this is 22 long, and don't you think they're the same? And, you know, in my 22 is the number of letters in the Hebrew language, and so, you know, I used to bring people in and say, well, it's a magic number, and there's a whole ideology around 22 in sort of ancient Judaism. The people in my lab were like, is he crazy? And the answer is yes. And so here's Dicer. It's, you know, it's sort of course it should interact with RNA. It's got an RNase 3 domain, and those trim off from the 3' end of a message. And so that's involved in production of siRNAs. And then this is what Craig discovered, RDE1, and then it turned out that RDE1, so this is a program that we developed in my lab, I'll tell you about it in a second, where we take the protein domains of RDE1 and we do a blast P against everything in the animal, many things in the animal kingdom, many things in yeast, many plants, many protists, sort of 500 across. So blue means there's a homologue, and a good one. This isn't weak homology. Bright blue is like, yeah, you'd believe it if you looked at it. And so you can see that the RDE1, Pee-wee, is found all across the animals. There's a couple of weird guys that don't have it. And then what was really important in the history of our work is that these proteins, the Pee-wee proteins, of which then these are all the worm paralogs. So these are different Pee-wee proteins. These are the ones Victor mentioned, ALG1, ALG2. These are the ones that mediate how microRNAs work. These other ones mediate other RNAi pathways. And the worms are sort of lunatics about Pee-wee proteins. They have 24 of them. We humans have 8. Plants have 12, 15 sometimes. Worms are really crazy on them. But what was really important here is that Saccharomyces cerevisiae doesn't have this, and that saved our asses, because if the yeast geneticists had been able to work on RNAi, they would have eaten our lunch. You know, they're just so damn good. And so thank God. But it's also interesting, why did these clades of yeast choose to delete it? And I won't get into it, but they dumped 99% of their introns. So yeast they have 100 introns left, and we have 100,000 introns. And so it's part of their trimming down of their genome. Whereas Saccharomyces pombe, Schizosaccharomyces pombe, has argonaute proteins and does RNAi. And it's not a coincidence that RNAi was discovered almost coincidentally in plants that have the argonaute proteins and fungi that have it, as well as in nematodes. But even more so, and this is the program, if you want to use it, you can just type into Google DepCod, and this was developed by Yuval in my lab and Ruslan, who's a collaborator at Mass General Hospital. So now we're going to go into using feeding RNAi, and you know, the discovery of RNAi by Mello and Fire was super important, but the discovery of feeding RNAi made it easy for all the rest of us to do it. Instead of having to inject double-stranded RNA, Julie Ehringer's group at the Medical Research Council figured out, and it was Lisa Timmons in Andy Fire's group who actually figured this out, that you could put in any worm gene, between a T7 RNA polymerase promoter, so you make double-stranded RNA in a bacteria and induced by IPTG, and you have to be really old to know what IPTG is, but some of you know. And that will induce this double-stranded RNA in the E. coli. The worm eats it, and the double-stranded RNA goes from the gut to the rest of the animal, and RNAi works. If you propose this in a grant, he'd kick your ass off no way. But it works really well. And so they made these libraries and sent it to us in around 2004. And so we could then just feed, and I'll show you what it looks like in a bit. And so we did feeding RNAi, and this was first with Craig Mello's lab, where we could take dicer and do RNAi on dicer, and it looks just like a LET7. Or we could take ALG1 or ALG2 and do RNAi, it looks just like a LET7. That's not enough to say it's the same, but we could also look at what happens to the Lin4 microRNA. So this is what it looks like in wild type if you're not feeding it anything, but if you get rid of dicer, now you have less of the mature and you have the precursor accumulating. And the same thing with Argonaute-1 and all the other peewee proteins here don't have anything to do with microRNAs. So just these two have something to do with microRNAs. And it worked on a second microRNA. It's not just Lin4, we could use it on any of them and they would accumulate a precursor. So one last thing to talk about RNAi is that there's a weirdism of RNAi in some organisms. So the RNAi that was studied in Drosophila or in humans, like us, is an RNAi that uses dicer and produces siRNAs that are the same length as the microRNA. But then there's a secondary amplification process called RNA-dependent RNA polymerase. And that takes a primary siRNA that's produced by this pathway, uses it as a primer in the same way that a viral replication works by priming, in the case of a virus, from a tRNA. But this is an RDRP that makes copies, and you end up with 1,000-fold more siRNAs if you have an RDRP. And worms turn out to have 4 of those, and one of them came out of a screen for enhanced RNAi that I'll tell you more about. And the other ones are important for sort of making RNAi work, so that if you inactivate this one with a mutation, we didn't do this, it was Ron Plasturk's lab that did it. They showed that this was important for somatic RNAi and the other for germline RNAi. So this is looking at the pattern of who has it and who doesn't. So this is taking the RDRPs, and what DEPCOD does is it says, okay, I got a pattern of blue and white. Who else has that pattern of blue and white? And so all the RDRPs look the same. And then ammonium transporters are sort of cousins, and maybe they act in the same pathway. And phosphoglycerate mutase looks similar, and maybe that acts in the same pathway. I'm not going to go over that. What I'm going to go over here is blue is who has the RDRP. So this blue, bright blue, is the worm finding itself. And all these other blues are animals that have RDRPs. I've told you that plants have them, and they use them to extend from the microRNA. But a whole bunch of animals like us don't have the RDRPs. And it's, again, not a coincidence that RNAi was discovered in the organisms that have this 2-stage. So it was discovered in plants, in fungi, in animals. But what are the animals that have it? So, this is an arachnid. This is a spider. I'll show you a list. So again, this is our DEPCOD way of looking at it, but you can also just do classic BLAST-P, and most people don't know, but when you do BLAST-P, if you go to the taxonomy tab, it will tell you who has it in order of how homologous it is. So the top hit, and here I'm excluding worms, so the top hit are these sort of mud-dwelling protochordates, so that they have the best homolog. And then we get down to scorpions and spiders and mites, and you know, it's like you're 13 years old and it's just what you want to read about is spiders and ticks and mites and they're badass creatures. And so what it means is that our worms are a lot more badass than we realized, right? And that we should respect their badassedness. And of the animals, it's you know, again, horseshoe crabs, and what do they have in common? They tend to go hypoxic a lot. So like the bivalves, you know, when the tide goes out, they close up, and they're hypoxic during that time. And plants have this, and plants spend a lot of time hypoxic because they get flooded when you water them, right? And I have no idea why that has anything to do with antiviral defense, but I'll bet it does, maybe, or not. Okay, so I'm going to end with about 10 minutes on, and how am I doing for time? Oh yeah, I got 6. Okay, that's approximately the same. This is a screen that Sylvia Fisher did. Now, I've told you that worms are really good at RNAi. We can do feeding RNAi. What a gift. But we still thought we could do screens to make RNAi better. Can we get mutants that have RNAi that's even more than normal? Which is kind of loony because it's so good. But another view of this is that RNAi is an antiviral defense, and maybe animals sort of induce or don't induce their antiviral defense depending on whether they feel like they're under a viral attack. And so maybe if we go for mutants that are enhanced for RNAi, we're putting the animal in an artificial state that would normally be very transient. It will only happen if there's a viral infection. But now, because of a mutant, we're putting them in a state much more consistent all the time. And so that's why it's sort of not stupid to go for things that are improved. You know, the normal way you learn genetics is this is all the ways to break an organism. But really, the power of genetics is it's all the ways to switch things on that shouldn't be on, or switch things off that shouldn't be off. And that's what physiology is all about. It's all about, you know, when do things get induced and not induced. And so these screening for enhanced RNAi is sort of trying to get at that, and I'll tell you why and how and why. So we start with a transgene that's a collagen, and it makes the animals roll on a plate. So they crawl around and they do these little circles, and if you don't do RNAi, this strain is rolling just fine. And if you have a mutant that makes RNAi work better, and this happened to be an Aries 6-7 that was one of the hits, now the animals crawl away. And so you could just do a screen for things that aren't rolling. And so out of that screen, Sylvia did it, and these are the hits. And she gets sort of 70 hits out of this, and this is in order of who's best. And the ones I'm going to tell you about are Lin35, Lin9, Lin54, but there's others as well. But these are the ones that are sort of fun. And she's now at Harvard Medical School studying these too. So these are some of the hits. And for example, these are called the SynMov-Bs, and I'll tell you about these in a bit. So if you take a particular double-stranded RNA that doesn't work very well in wild type, it barely causes lethality. We don't know why, but we know that in these mutants that are in what are called the SynMov-Bs, now RNAi works way better, and we get the lethal phenotype almost 100%. And what are the SynMov-Bs? So these are mutations that were originally identified in the Horvitz lab. I'll tell you a little bit about them. And so a wild-type animal will make this vulval protrusion that's involved in mating with males to then fertilize the eggs. And that's normally made with one. And the way it makes one is by an anchor cell that's adjacent to this hypoderm. sending a signal. It's a growth factor signal. And so they were interested in this because this is a RAS signaling pathway. But the mutants that they got, and these mutants were presented by Chip Ferguson in Horvitz lab group meetings when Victor and I were graduate students, and we would hear all about SynMov-A's and SynMov-B's, because the original mutant that was isolated was a SynMov-A, SynMov-V double, and it caused this extra MOV. And then the Horvitz lab did amazing work over about 10 years, 10 different papers, showing that you need to have both of them knocked out. And what that leads to is too much of this growth factor from the hypoderm, not from the anchor cell. And it's kind of just weird stuff. But then we get all the SynMov-Bs are enhanced for RNAi. And what does that have to do with this? We'll get to that in a minute. And there's Bob, and this is our Victor and my mentor. And this SynMov work is sort of number 8 on his list of, what people will say major discoveries, but it's utterly awesome stuff, wonderful papers. And of course, Bob has been important in our early work, but it's coming back. This is all in the last 2 years, right? So what should I work on next, Bob? Please. So what do these turn out to be? So Lin35 is the gene that's sort of the most sexy, and that's retinoblastoma, and that's an oncogene that's involved in retinal tumors, one of the very early oncogenes. But there's human homologs of Lin9, Lin37, all these things have homologs, and what these do is they sort of assemble onto what's called a dream complex. And I had originally kind of resisted working on this because it's sort of chromatin regulation. I was really worried that I will never make sense of chromatin. Others can, but I wouldn't. But we've gotten more and more interested, and I'll show you why. And this was the experiment that really made it fun. So we started with a Lin8 mutation. So that's a SynMov-A. It has no phenotype on its own. And normally, in order to get it to show a MOV phenotype, you have to do something to knock down a SynMov-B. And we decided, well, gee, we know that if we knock down a SynMov-B, the animal is enhanced for RNAi. And so maybe if we do a virus infection, so the Orsay virus was identified by Marianne Felix from an orchard in France, and so you could infect a worm with Orsay and ask, if we take a Lin8 mutant and infect it with Orsay, do we get a MOV phenotype? And so if we take that Lin8 mutant and don't infect it, nobody's MOV. But if we take that Lin8 mutant and infect it in 3 separate replicates, we basically get 10%, 10%, 10%. And so that says that a viral infection essentially downregulates SynMov-B somewhere. And this is what a SynMov-A, SynMov-V double, it's 100% MOV. And so the idea is during a viral infection, this SynMov is being turned down, and that helps the animal resist the viral infection because RNAi is enhanced, and that's what you want during a viral infection. You want to be resistant to a virus. And in fact, we could see it when we look at the amount of RNA by RT-PCR. So a wild type has sort of this much, 1, and a Lin35 mutant that's constitutively airy resists the infection much better. It's about 20-fold better. And a Lin15B, which is another SynMov-B, resisted about 100-fold. So it says that this is a natural state, and the reason they're airy is because we're constitutively putting the animal in an antiviral state. And antiviral states are important. It would have been better during COVID if humans were in an antiviral state. The animal loss of the RDRP seems like a bit of a mistake. Maybe it's because we have a nervous system, because it seems like plants didn't lose their RNA-dependent RNA polymerases. Okay, I'm going to skip this last bit. And I want to go to talking about the people in my lab. So this is the lab today at our house. And the work I talked about just now is from Ashwin and Himani. And there's other projects I didn't go over. And this is the list of people in the lab over the years. And I want to mention, of course, there's a lot of names. I won't go through all of them. But one of the things I want to mention is, you know, lots of times I go and I give a talk, and they say, talk to us about mentoring. And my lab has about 800 person years of people. So, you know, most of these people were in the lab for 6 years approximately. And so they, you know, multiply 6 times the number of people, and it's 800 person years. I'm 40 person years. And even if I say, oh, I'm really good, so maybe I'm worth 80 of those 800, that's still 10% of the lab. And when people leave my lab, there's a hole, right? We lose talent and knowledge, and the people who spent time in my lab remember each other a lot and are friends forever, and they write papers together 20 years later. You know, it's just the most wonderful thing to see. So the mentoring in a lab is the lab. And you know, yes, I'm an important part of it, but I'm not at all the most important part. And I want to mention Diane Sacchetti, who has been the soul of the lab for 26 years and has been really important in all the people. She cares a lot about all the people. And many of you might have seen the famous ice cream truck scene that's on Twitter, and I don't know, got 1 million hits of, you know, me the day of the Nobel announcement serving ice cream. It's because her husband, Frank, who's the family friends, has an ice cream truck. And of course, he came on October 7th with the announcement of the Nobel, and we served ice cream to everybody at Massachusetts General Hospital. Super fun. And this is the home. There's Natasha, my wife, at the Rolling Stones concert, at the famous Food and Books. She's been the key part of the lab, my life. And my lab has always had a theory that she's the brains of the operation, and it's true. I learn a lot from Natasha. Our daughter, Victoria, there she is in medical school, and it turns out there's an instinct to be excited when your kid's a doctor. It's really exciting. And Victoria's been the light of our life, and there she is getting her degree from Amherst, and Natasha was able to give her her degree because Natasha teaches at Amherst College, history of art. And there's Victoria with our dog Barnaby, who we talk to more than anyone in the house, in a high voice, a lot. I wanted to thank my parents. My dad went to the University of California, which is why I went to the University of California, as did almost all my cousins. And he was an engineer for many years, and that really taught me how to think about technology. And I sort of I didn't become an engineer, but I could have, should have. And my mother did not go directly to college. She came out of high school and went with her sister to the Young Hebrew Women's, like the equivalent of the YWCA in the old days. And that was where a virtuous girl at age 17 in her case, and she went to the San Francisco Young Hebrew Women's, whatever it was called, and met my father's sister, who met my father, and one thing led to another. And we did our homework together, my mother and I. That's me at a science fair project. I blame my parents for the terrible orbital mechanics of all the planets being in one orbit. This is inexcusable, but I was only 6. And this is my early work was becoming an electronics thing. That's what nerds did before the internet, before computer programming. And I learned electronics, and it really taught me a lot. And then just a quick sort of, this is going backwards in time, lab and home, important. Harvard and MIT, important. I spent a year on the road in South America. That taught me about surprises and gave me 100 stories to regale people with. And they might, it seems like after 6 years they get tired of them and leave. But it works for 5 or so. And then I lived in my van for a year at Berkeley. I did physics, and what that taught me were scientific revolutions. And not that I participated in them, but I got to read about them. And physics is taught historically, which is really important because it teaches you sort of the temporal sequence of how things are learned, and it sort of prepares you for revolutions. And when I got to Harvard in 1976, it was obvious that the recombinant DNA was a revolution. And now with genomics, that's the result of that revolution. And it has been a revolution for me, for sure, and many of us. And it really all started with space program. That was propaganda to tell the Soviet Union that we were going to win or send missiles towards them. But what it really did was inspire a whole bunch of nerds of my generation to become scientists. And that's why we have Silicon Valley in the US, and why we now have a good pharma industry and good biotech, and a whole lot of us were inspired by that. Lastly, as a kid, you sort of say, well, who am I like? And so I sort of bonded with all the people on TV. And so the early TV and radio was very Jewish. And as a Jewish kid, I said, maybe that's like me. And so I wanted to be funny. And so I try. And but I think funny is not just for comedy. I think funny is a way to look at the world at a slightly different angle, and that's very scientific. And so I actually think one should use that as a criteria for selecting postdocs. How funny are you? And I think that's it. Oh, sorry, Fred. Asabel I want to thank. That's how I started in molecular biology. That was my PhD advisor. He worked next door to me at Massachusetts General Hospital for many years. And Scott Kennedy, who was a postdoc, gave me a deer head that he shot. And Fred was in the office behind me, and I tried like hell for many years to find a deer butt to put in his office, because I thought it was appropriate for somebody who's your former boss. But you can't do it with like a junior faculty member. That's abusive. But I never did find a deer butt, but I always looked. Wally Gilbert was just this luminous figure in the Harvard biolabs that sort of a role model of how to be a scientist. And there were many people like him at Harvard that really inspired me, and over the years sort of taught many of us how to be scientists. And lastly, my department was funded by Hoechst Pharmaceutical Company because of the work of Hans-Hermann Schörne and Ernst Winokur. I'm actually the Hans-Hermann Schörne investigator at Mass General Hospital. And that was because Howard Goodman had cloned insulin. They produced insulin, and they thought maybe they could have that they could support it. And it was really because Dr. Schörne had a lot of pro-American feelings after World War II, where he had been a 15-year-old soldier in the army in Germany, and was treated well by the US during the post-war period. And so I was a recipient of, World War II didn't end at the time that I was working in it, and it led to wonderful support in my department. So thank you very much.
Dear guests, the 2024 Nobel Lectures in Physiology or Medicine have now come to an end. I would like to extend our gratitude to the Nobel laureates for their extensive presentations this afternoon. Thank you all for attending, and thank you for sharing this significant moment with us. When you leave the hall, please leave the same way you came in. Those of you continuing to the reception, please be prepared to present your ticket. The QR code will be scanned. And now there is an opportunity to photograph the Nobel laureates on stage. May I ask the laureates onto the stage for a round of applause?